Hello. Um, I'm Jeff Way, and I'm going to be talking about recoding the Salmonella genome. And this is on behalf of a number of people who actually did most of the work. And one thing I want to comment here, this is doubling as an acknowledgement slide, but I don't want you to be intimidated and think that this is how many people it really takes to engage in genome recoding. The first three people did most of the wet lab work, and then many of the rest of the people were either involved in the design aspect, and they were people rotating in and out doing software design and so forth. And then there was an excessive amount of middle and upper management. And so it really is not that bad. The money problem is more of a serious issue than the personnel, I think. Okay, so why salmonella and why recoding? The reason for salmonella is as follows. Basically, what we really want to do is use salmonella as a live vaccination vehicle. Right now, when you get vaccines, there are often attenuated viruses that are live viruses that serve as vaccines. You really cannot do that with confidence with bacteria, because bacteria can go into your gut or get into whatever the environment they're in and mix with other bacteria, and they exchange genetic information all the time. So if you attenuate a bacterium, it can easily recombine with something else in your own microbiome and become non-attenuated. And so what we really want to do is create a situation where horizontal gene transfer is blocked. And so part of the idea here is that we could do that by recoding the genome, dropping out some codons, and then incoming DNA cannot be translated. Salmonella specifically is very useful as a live vaccination vehicle because it induces many of the immune responses that you want with respect to cell invasion leading to antigen presentation and so forth. It's really kind of the perfect bacterium for vaccines that would protect against this family of bacteria, which includes EHEC and Shigella, and also Shigella dysenteriae, for example. And Salmonella LT2 specifically is only a mild pathogen. It is somewhat toxic to mice, but really not to humans. And I've heard anecdotal stories about people in the old days actually, for example, doing Salmonella biochemistry, sucking pellets up and down to resuspend them and actually swallowing 10 to the 11th bacteria and not really maybe feeling too good for a few days, but nothing that serious happened. And also, while it is not E. coli, so we're doing something novel, all the same genetic tools basically apply. Okay, so the design specifics that we settled on were to only change two codons. We changed the two leucine codons, the kind of oddball leucine codons, to more the set of 4. CTN is a leucine codon. And so this is all we did, 2 codons out of the possible 64. And I do have to acknowledge George Church in his lab. Neely Ostrov gave a kind of an introduction to the project and their work on E. coli. And early on in this project, we did talk to George and some members of his lab, and they talked about some of the problems that they had, and so the real practical issues. So our project was designed in part to address some of the practical issues. One of the practical issues that we were worried about, since we want to have something that could ultimately be given to humans, is that we want it to be fairly healthy, and so we were worried that changing 6 or 7 codons would have a deleterious effect on growth, and so we just limited ourselves to enough that should be sufficient to prevent horizontal gene transfer, but not so many that we were really just changing a lot. Also, we removed in our design the restriction system of Salmonella, and we knocked this out first. There are 4 prophages of about 50 kilobases each, numerous pathogenicity islands, a few pseudogenes, a few transposons, and all of this totaled about 400 kilobase out of the 4.8 megabase starting genome. So that's just that much less that we have to make, for one thing. Also, one restriction site, this BSPQ1, is completely removed, and this is a favorite restriction site for all sorts of manipulations because it recognizes a sequence and then cuts adjacent to the sequence, and so you can use it to cut off linkers and PCR ends and so forth. We used a stepwise integration strategy to, and I think there's nothing, this is kind of obvious to anybody who's done bacterial genetics, that this is what you would want to do. The idea is that you put in a new piece of DNA, and at the end, at the junction between new and old, you have a drug resistance marker, and then when you put in your new linear fragment, you have a different drug resistance marker. You have a recombination event happening out here, and you select for this one to come in, and then you screen among those for recombination events that happen out here and get rid of the old marker, and so you can use this over and over again. Okay, the particular design challenge that I will mention is there are a number of cases where there's overlapping genes, and what we did use was a piece of software generated at the Venter Institute, which predicts the functional effect of amino acid substitutions. Basically, if we want to make a substitution here and there's an overlapping gene, we are often forced to introduce a sense change, and this software helped us figure out what to do. Okay, the whole method is called SIRCAS, maybe named after the Showtime TV show about the political system that you may have watched. The basic idea is to synthesize small pieces of DNA by chemical means, and then assemble them in yeast using its recombination features to make a circular chromosome, and then amplify that by rolling circle amplification, and then digest and then finally linearize the DNA with this enzyme. This is the same as BSPQ1, to give us linear fragments of 10 to 25 megabases, and then we recombine those into E. coli, and we actually amp up the recombination system using lambda red, which works as well in Salmonella as it does in E. coli. I'll skip that. That just says that this counter screening idea works. And finally, the progress thus far is that we've done over 200 kilobases. Of the 1,500, the whole design involved over 30,000 changes. We successfully instituted that many, and in the section that involves sequence-verified DNA, we had only a very small number of mutations, but in cases where we used non-clonal DNA fragments, we had quite a number of mutations, and that's really not viable going forward. Okay, so the key advantage of this method really is that it's 2 days per integration cycle, and the lethal and deleterious mutations that we introduce by design, and we don't realize that they're lethal or deleterious, become immediately obvious, and we only have to look through a small region to identify them. Okay. I'm getting signs that I'm supposed to start the question and answer period, so thanks to the people who did all the work, and also to DARPA BRICS. The Wyss Institute funded us, and also various groups synthesized DNA for us. Thank you. Questions? Come on. Oh, yes.
How many bad codons so far among those two? How many instances, if any, in the 200 kb that you've tested have proven to be unchangeable?
We did not find any so far. It is possible, again, comparing our effort to the Church Lab effort, it's possible that they picked tougher regions. Also, but the main difference might be that they were changing 6 or 7 codons, really, and we were only changing 2. That's why we chose to only change 2, is we wanted to avoid that problem.
How long long a fragment are you integrating, Go?
10 to 25 kb. We did some at 10 and some at 25. Basically reasoning that if there was a problem, we wanted to find it immediately and be able to detect it by sequence gazing.
One of the aims of the codon changing in these projects is to create like a genetic firewall.
Correct.
Will your Salmonella be firewalled against George's E. coli? Or have you both now got compatible organisms?
That's a good question. I think if their organism sent DNA into ours, it would be okay. If our organism sent DNA into theirs, it would not work because they've changed more codons. I think there was one more question, maybe? Maybe not. All right, maybe running out of time.